This video will focus on the regulation of gene expression in bacteria. So we'll be asking the basic question, how are genes expressed only when needed? And we're going to focus on transcriptional control as it is the primary site at which gene expression is controlled in bacteria. We're going to initially focus on a system whereby bacteria can metabolize or break down lactose and use it as a source of energy. So lactose, of course, is just a simple sugar. And I just want to point out from the get-go that there are a couple of genes involved in lactose metabolism that have to be expressed in order to metabolize lactose. So toward the bottom, we're looking at a schematic of a bacterial cell. And here we have lactose entering into the cell, so passing through the cell membrane, through this green protein right here. That's a permease. So there is a gene, the LAC-Y gene, that codes for that protein permease. Once inside the cell, lactose is broken down by an enzyme, beta-galactosidase, into two individual sugar molecules, namely glucose and galactose. So that enzyme, beta-galactosidase, is coded by the gene LAC-Z. So we're going to be focused on the expression of these two genes, the LAC-Z and the LAC-Y, again being expressed when lactose is present, but not expressed in those genes when lactose is not present. So I also point out that yes, once inside the cell, lactose is broken down into glucose and galactose, but it can also be modified into allolactose, so just another form or another version of lactose. If I give you a couple of scenarios like this and ask you to predict whether or not the LAC genes would be expressed, you should think about these logically and think, okay, number one, if you're growing the cells on glucose only, well, probably the LAC genes would not be expressed. No need to express those genes if you're only given glucose as a food source. If you're grown on glucose and lactose, you might logically think, yeah, sure, those LAC genes probably are expressed because lactose is present. If you're given lactose only, then sure, it would make sense to have those genes expressed. So you might look at these and think, yeah, conditions 2 and 3 would probably result in the LAC genes being expressed. But it turns out that the LAC genes are not expressed in the first scenario, but they're also not expressed in the second scenario either. But they are of course expressed in the third scenario. So this tells us two things. First of all, cells can monitor and detect when they're given lactose, and they can express the LAC genes in the presence of lactose, but they also preferentially metabolize glucose. So if glucose is present, they won't express the LAC genes, at least not to a very significant extent. So let's explore this gene regulation system a bit more, beginning with some basic concepts. So what we're looking at here toward the bottom are some colored sketches. So the colored bars represent DNA. They represent part of an operon. We'll talk about operons in a bit. But what we have here toward the left is an illustration of positive regulation. What's going on here? Well, when genes are regulated in a positive fashion, there are activator proteins, we see one right here, that bind to certain regions of DNA. When they bind, that then facilitates the binding of RNA polymerase to the promoter, which then would allow our genes in question, in this case so far we might be talking about the LAC genes, for example, but allows those genes to be transcribed. If the activator is not present, RNA polymerase cannot bind to the promoter, and as a result, our genes, shown here in green, are not transcribed. On the other hand, other genes under the control of negative regulation, shown toward the right, that system involves a repressor protein, which binds to the operator, which again is just a portion of DNA. When the repressor protein binds to the operator, RNA polymerase cannot bind to the promoter, or even if it does, it can't make its way past this region right here, and as a result, our genes, again shown in green, are not transcribed. If the repressor protein is not bound to the operator, RNA polymerase can bind to the promoter, and as such, our genes are transcribed. So that's just illustrating the differences between positive and negative regulation. So what is it that determines whether or not those activator proteins or repressor proteins will bind to DNA? Well, in a general sense, we can talk about allosteric effectors. So we're just talking about molecules that can bind to either activator or repressor proteins, and when they do, they cause them to change shape. So as one example, if you notice toward the bottom here, we have a repressor protein bound to DNA, bound to the operator, But if lactose or allolactose is present, shown by the small red structure here, that can bind directly to the repressor protein, causing it to change shape. As such, it does not bind to DNA, and the genes downstream can be transcribed. So let's talk a bit more detail about the lactose system. So we mentioned earlier that there are a couple of genes, the LAC-Z and the LAC-Y. There's also a third gene, the LAC-A. We're not going to focus on that too much, but these genes are coordinately expressed when they need to be. So what we're looking at here is this concept of an operon. So multiple genes, Z, Y, and A as well, under the control of a single promoter. That's what the P refers to right here. So we're talking about an operon. This is what we're talking about again. Multiple genes under the control of a single regulatory region. So let's explore the lactose system in a little bit more detail and look at regulation of the LAC operon. Your book has a figure that illustrates the regulation of the LAC operon in the presence and absence of lactose. In class, I drew a picture that looked like this. So let's just quickly walk through this. So again, we're looking at part of a bacterial chromosome. So this is DNA, this line here. Some of the major regions involved here. First of all, this I region. We'll come back to its role in just a second. Next, we have our, I should mention real quick, the I region actually is not part of the LAC operon. It's sort of upstream and encodes for a repressor protein, but we're including it here because it plays a big role in expression of our LAC-Z and LAC-Y genes. Next, we have a promoter, an operator, and then we have our structural genes, namely LAC-Z and LAC-Y. And again, there's that third one as well, but we're not going to focus on LAC-A. What happens here? The LAC-I region codes for a repressor protein. If there's no lactose present, the repressor protein has a shape that allows it to bind to the operator. When it's bound to the operator, RNA polymerase, even if it binds to the promoter, it can't make its way downstream, and there, as a result, our LAC-Z and LAC-Y genes are not transcribed. So again, that's in the absence of lactose, right? Our genes are not transcribed. In the presence of lactose, something different happens. Here we have lactose indicated by a triangle. Turns out lactose can bind directly to that repressor protein. When it does so, it causes that protein to change shape. In this new shape now, so it's been allosterically changed, right? Its shape has changed. In this shape, it does not bind to the operator. As a result, RNA polymerase can bind to the promoter, and our genes would be transcribed. And that makes sense because you want your genes to be transcribed when lactose is present so that the cell can metabolize lactose. Notice we didn't say much about this part of the promoter, this CAP binding site. So CAP refers to a catabolite activator protein. So this is the site on DNA where the catabolite activator protein can bind. So that's another protein involved in the cell system. We'll talk about that in a second. And also, here's a complex right here consisting of CAP as well as CAMP, or cyclic adenosine monophosphate, or CAMP. So what happens is that this CAP-CAMP complex can bind to the promoter. That then also facilitates the binding of RNA polymerase to the promoter, which would ultimately facilitate transcription of these genes. If this CAP-CAMP complex is not bound here, RNA polymerase tends not to bind to the promoter, and as such, our genes tend not to be transcribed. So how does this CAP-CAMP complex play into the whole system here? Well, this has a direct relationship to the amount of glucose available to the cell. So far we've focused on gene expression in the presence or absence of lactose as an example of negative regulation. So now let's talk about how glucose comes into play as an example of positive regulation. And so the whole phenomenon is referred to as catabolite repression. Recall that glucose is one of the breakdown products of lactose. So we could say that glucose is a catabolite of lactose metabolism. What happens when glucose levels are low, so there's very little glucose present, we end up producing a fairly high amount of CAMP, of CAMP in the cell. And those high levels of CAMP are needed to bind to catabolite activator proteins, which are also produced in the cells and bacteria. Those two molecules combine together to form a CAMP complex, which, as we said a few minutes ago, can then bind to the promoter, which again facilitates binding of RNA polymerase and expression of our genes. So again, low glucose levels, high CAMP levels, high levels of the CAMP complex that can bind to the promoter and facilitate gene expression. If high glucose levels are present in the cell, you tend not to have much CAMP produced. Therefore, you don't have much CAMP complex. Therefore, they don't bind to the promoter, and as such, the genes tend not to be expressed. And so that's how glucose can play a role in determining whether or not the lactose genes are expressed or not. Another concept you should be familiar with is this idea of cis-acting and trans-acting elements. So first of all, toward the top, let's focus on cis-acting elements. We might say that operators are examples of cis-acting elements. What does that mean? That means that operators play a direct impact on the expression of genes on the same chromosome. Okay, and hopefully that makes sense. So we sort of see that sort of illustrated here. On the other hand, we can talk about repressors or repressor proteins as being transacting. So in other words, we have a part of the bacterial chromosome that codes for a repressor protein. That repressor protein might have an impact on the same chromosome as the region at which the repressor protein was coded from. But it can also, again, think about it, it's a protein is floating in the cell, it can also impact other chromosomes. Right? So if you have two copies of lac operon, a single repressor protein formed on one chromosome can impact the operator on that same chromosome, or it can certainly have an impact on another chromosome in the cell as well. So that just illustrates the concept of cis versus trans-acting elements. Okay, so we talked about the lac operon. Let's now talk about another example of an operon. In this case, we're going to focus on the Arabinose operon. This it's similar to the lactose operon in that the structural genes, in this case the genes B, A, and D, these are expressed when arabinose is present so that the bacterial cells can metabolize arabinose as a food source. So some of the major regions involved, the C region, that's a control region. As we're going to see, that codes for a control protein, operator region, and initiator region, and a promoter region. And then our structural genes, BAD. So the regulation of this operon, of the ARA operon, goes something like this. When arabinose is present, a protein, a control protein, we abbreviate it ARAC, which comes about as a result of being coded for by the C region, that control protein binds to the I region, like we see here. And we also have the same CAMP complex that we discussed before with the lac operon. And as a result of this control protein binding to the I region, again, in the presence of arabinose, our structural genes, B and A and D, are transcribed. Okay, and again, this makes sense because these are genes that are involved in metabolizing arabinose. So in class, I probably drew a picture that looks something like this. So again, here's our DNA, part of the bacterial chromosome. Here's our control region, our operator, our initiator. Here's our promoter, and then our three structural genes. So the control region codes for an activator protein. We can abbreviate it ARAC. In the presence of arabinose, indicated by the small triangle here, the larger triangle, that's the actual ARAC protein. So in the presence of arabinose, this complex binds to the initiator. That facilitates binding of RNA polymerase, and as a result, our genes are transcribed. And again, we have the same CAP complex involved as well. So low glucose levels, you'll have this complex form. It will bind to this, in this case, the initiator, and again, facilitate gene expression. Okay, so that explains what happens when arabinose is present. What about when arabinose is not present? Well, again, in this operon, we have a system where a single protein, this ARAC activator protein, is involved in both positive and negative regulation. So when arabinose is absent, let's now focus on the bottom picture here, that control protein, that ARAC, still binds to the I region, but it also binds to the operator, as we see here. In this conformation, we have the single protein binding two different regions. And as a result, in this conformation, again, there's no Arabinose here, right? As a result, our structural genes are not transcribed. So here we have a system that displays both positive and negative regulation just by a single control protein. Okay, let's now focus on a third example of an operon, in this case the tryptophan operon. In this case, genes are expressed when bacteria need to synthesize or produce tryptophan. So the previous two operons, those are systems where bacteria break down or metabolize certain sugars. In this case, now we're talking about expressing enzymes involved in the synthesis of something, namely tryptophan, which is an amino acid. The way it works is similar to the lac operon, although sort of in reverse, as we'll see in just a minute. So in this case, the presence of tryptophan causes a repressor protein to bind to the operator and block expression of the genes. Again, as we'll see in just a second. So here we see schematically the tryptophan operon. So here's an operator. Our structural genes E, D, C, B, and A. Each of these codes for a different enzyme involved in the synthesis of tryptophan. And notice what's interesting is that the sequence of the genes tends to correlate with the sequence that the enzymes are used in the synthesis of tryptophan. So in class, I drew a sketch that looked something like this. So again, we're talking about part of bacterial chromosomes, that's DNA. We have a repressor region, promoter, operator. We also have this region here called the leader region, and then our 5 structural genes, E, D, C, B, and A. So what happens? The R region can code for a repressor protein. If there's no tryptophan present, the shape of the repressor protein is such that it does not bind to the operator. As a result, our structural genes are transcribed. If tryptophan is present, no sense in wasting time and energy expressing those genes. So what happens? Tryptophan, indicated by the small triangle here, binds to the repressor protein, causing it to change shape. In this new shape, now it binds to the operator. And even if RNA polymerase is bound to the promoter, it can't make its way downstream, and as such, this blocks the expression of our structural genes. So if tryptophan is present, genes are not transcribed. If there's no tryptophan present, then of course the genes are transcribed. But notice this leader region right here, the sequence of bases. We haven't said much about this yet. Let's talk about that leader region and the role that it plays in expressing the structural genes as part of the tryptophan operon. So although the repressor protein is the major player involved in regulating the expression of the tryptophan operon genes, there's also some fine-tuning that takes place as well by this process we refer to as attenuation. So what we see toward the bottom is a schematic of that leader sequence. So 5' to 3', all the bases there. And notice as part of that leader sequence, this region right here, these two codons, UGG, UGG, they both code for tryptophan, which of course is the amino acid that we're talking about synthesizing as part of this operon. It turns out that the structure of this leader sequence mRNA is such that it favors transcription of the tryptophan genes when tryptophan is low, and it tends to terminate transcription of those genes when tryptophan is high. So how does it do that? How does that all work? Well, toward the left we see schematically our leader sequence, and notice there are several regions where complementary base pairing can occur between parts of this mRNA. So regions 1, 2, 3, and 4, again, are regions where base pairing can occur. And what we can have is a situation where regions 1 and 2 compare together, 2 and 3 compare together, or 3 and 4 compare together. So why is that relevant? And how does it play a role in regulating the expression of the tryptophan genes? Again, just I'll quickly point out, if you see up here in red, UGG, UGG, so there are our two codons that code for the amino acid tryptophan. And again, what we're looking at here is messenger RNA. Toward the right, let's focus on the lower right portion initially. Again, we're talking about bacteria. So recall that translation, here we have a ribosome interacting with our mRNA. Translation can occur at the same time that this mRNA is being produced by transcription. right? So that's key to understanding how this works. So lower right, what we have is a situation as follows. Let's say the situation is such that tryptophan levels are low. So we need to synthesize or tryptophan, in other words, express those TRP genes as part of the TRP operon. So what we have here is a ribosome translating the mRNA, again, before the mRNA is even done being transcribed. So what happens if tryptophan levels are low? Recall that those two codons found as part of the mRNA that code for tryptophan will be encountered by the ribosome. If tryptophan levels are low, the ribosome will stall at that site. When that happens, that favors complementary base pairing between regions 2 and 3. As such, that then allows transcription to continue. So again, our structure genes would be farther to the right here. They would be downstream. Those genes can then be transcribed. On the other hand, if we focus toward the upper panel, when tryptophan levels are high to begin with, the ribosome will encounter those two tryptophan codons and it'll zip right by them and no problem because there's plenty of tryptophan available. It won't stall at that site. And so as such, region 2 will enter quickly and directly into the ribosome. When that happens, that favors complementary base pairing between regions 3 and 4. That forms a hairpin loop that tends to terminate transcription. So in other words, so this is just the leader sequence of the mRNA, before this is even done being transcribed, in other words, our structural genes, which would be farther to the right, transcription is terminated. It's stopped. And so again, that makes sense. If tryptophan levels are high, no sense expressing those tryptophan genes. So this just illustrates the mechanism by which we think that process of attenuation is involved in regulating gene expression in the TRP operon. So this concludes our brief discussion of gene expression in bacteria.